on record my thanks to Dr. Gitanjali for having invited me to this workshop. Morning. So it was the thought of the angel and she came up. So I was actually putting on record the thanks to Dr. Gitanjali for having invited me. Thank you very much. So yeah, I mean, this picture in the background, since I am going to talk with reference to how this data liberation becomes important when you have some long-term studies planned or already doing, and when you are actually choked at some point in time for want of certain things. So how this data liberation becomes so important and profoundly inevitable rather. This picture actually is one of our sites, a grassland site in the Kashmir Himalaya that is part of the long-term ecological observatory network of the country that MOEF set up, I think, in 2019. And Ishrat is a student who actually was the first PhD student who started this. Anyway, so how do we move slides? Okay, so when I looked at the title of this workshop, Liberating Data, it sounded fascinating to me, and I thought what exactly this liberation means. Let me read something about it. And there was a book, by this Birch and Cobb that talks about the liberation of life. And it's a fantastic book. It's very openly available. You can Google it out. What exactly this liberation means in life. You come out of oppression, your tendency of being free, so on and so forth. And then it is written in an ecological context. Largely, I mean, it's a very nice read that way. And how basically it is so that there's a lot of data that exists there, but we do not somehow know it or have access to it. Or then, since this book is written in a different context, I got just at this moment something in my mind, which is a, it's a kind of poem by Iqbal probably. It says that जहां में दानिश व बीनिश की इस दर्जा है अरजानी, कोई शय छुप नहीं सकती, ये आलम है नूरानी, ये दुनिया दावती दीदार है फर्जंदे आदम को, ये हर मस्तूर को बख्शा है ज़ौक़ उरियानी. यानी कि it says that everything in this world, in this universe that is in the closet or that appears to be in the closet has the inherent tendency to come out of that closet. The only thing is that we need to scratch a bit. And I believe this workshop is a fantastic exercise in that, direction. Well, I believe that we deal with a situation what I call as data paradox as well. That a paradox in the sense that we have two situations. One that we all know that data is power, of course, yet we deal with a lot of issues of data blindness. Many people do not know what this data is and how to use it. And this data liberation therefore becomes important. And since I call it a paradox because there is a lot of data that doesn't need any liberation, but is still very much underutilized, not utilized at all. That's where you need this data mining and you need data synthesis without that liberation effort. And on the other hand, there is a lot of data that we badly need for asking many questions at different scales, but that's locked up either in the, say, museums, the old manuscripts, herbarium specimens, old scan documents, so on and so forth. So how do we navigate through these situations? And in fact, there are, they say, more than around 400 million specimens housed in different herbaria. And many million pages of biodiversity literature that are not currently machine readable, as you might have been kind of given to understand since yesterday as well. So how to basically deal with this situation, how to get it out. And we all know that this tools like AI and machine learning and deep learning that are coming now, all sorts of these deep seek and other things, they are helping us a great deal. And if you look at this annual growth in the number of publications that cite AI and the machine learning applications, for example, there is a dramatic increase since 2018, 20, in the publications as such. And what we can use basically these AI tools probably for, both for generating quality data and of course liberating the data that is existing but we do not have access to. And in addition to that, all sorts of applications that we see, instant species identification, automation of biodiversity science, monitoring habitats, you can add everything. Most importantly, I see its merit in trait ecology. When you deal with the trait data in the databases like say try, give, it's to begin with seems like a mess how to get your things done. So these AI tools can be really important in that direction. Well, let me begin with some of the difficulties that I faced long back, I think 2010 or 2011 when I started working with herbarium specimens. We tried to, since one of the major focus over the past few decades has been plant invasions, and we do different stuff with the invasive species. One of the questions that occurred to us long back was that what's it that leads to the shift in the ecological fortune of species when they move from their native to the non-native range? What happens to them? I mean, on what basis can we predict their distribution, right? And the fundamental question that we asked at that time was that probably a species that has a wider native range, if those species go to a non-native range, they have more probability of becoming invasive. Why? Because they have a set of traits which make them kind of more abundant in their native range, which predispose them to be more invasive because of those traits. So we set on to kind of answer this question, and we use two things. These native species that are, European native species, because from Europe has kind of delivered or exported these species all across. Our country, for that matter, having that colonial past, has many species came, connected to that history. And of course, from Europe to the North America also, there have a large number of species that have been migrated, right? So our issue was that, is it really so that the number of, the abundance of a species in its native range would predict its degree of invasiveness in the introduced range? We had two situations. For Kashmir Himalaya, we somehow managed through some of the resources, but we had to deal with more than 800 species that were European native and invasive in Canada and that not part of North America. Believe me, for this study, I had to work physically in many herbaria of North America, although still that time some degree of digitization of the data was there. But it was a Herculean task for me to deal with that situation. But what we found, what I'm trying to tell you is that what we found at the end of the day, yes, there is a clear evidence that those species which are having a broader native, because we got that all these phylogenetic perspective and of course the climatic perspective into the models, and we found that yes, it's true that yes, it happens. So we have evidence both for European species that have invaded Himalayas and European species that have invaded North America, which was a learning from that exercise. But over the period of time, if you look at this, since 2010 onwards, 2012 onwards, there has been a lot of improvement in terms, a lot of effort actually driven by the GBIF and many other global initiatives of digitization of this herbarium specimens. And you have now a lot of information easily accessible. So with at your desk, you can access that very valuable information, because I believe these herbarium specimens are a very, very important source of information, invaluable information. And much of it is not actually used so far. So this is what makes it a bit easier now. If we have the appropriate tools to use this data that is now existing or that is now getting more and more available, it's a great deal. But in the Indian context, I still believe that our herbarium data is not digitized to that extent. And an effort, I mean, we have to kind of pursue this with the concerned, say, BSI and other agencies to have it as a national effort, so that nationally we will have at least access to this important data from that perspective. And one of my colleagues, this Christophe Rocher, he has been a collaborator for last now more than a decade. We did an Indo-German project together. He did it recently with this Centauria, and they published this study just a few months back in the New Phytologist, where they found that, using the herbarium specimens, how this Centauria stabi, it has diploids and the tetraploids, as I was telling you, how they basically have invaded particular regions, not the entire range, and how basically they can link it to the population genetic stuff of this species, and drawing very robust generalizations. Now, since AI tools are now coming in a big way, and these AI tools can be of really immense help in dealing with the herbarium data. When you deal with the herbarium data physically, you come across many challenges. First, many specimens are torn out, labels are not so legible. Many traits in the specimen you can not figure out easily, right? And in that context, what these AI tools can help us with is that, much of the information which is otherwise not readable, it can become readable. There are different tools for that matter. And then many of the traits that have never come to our mind actually could be important in different contexts. Those traits we can figure out through these, for example, like the typical small petiole length at times. Say sometimes these specimens are herbivore damaged, right? This extent of herbivore is a very important ecological trait that we can infer from. We did some exercise recently with a species called Coneza. The question was that how this species across its global range of occurrence varies in the degree of herbivory. The premise was that it should have less herbivory in the non-native range because of the release of enemy release, hypothesis. And we had a very good large number of, I would say, regions across the world. I don't have the data here. I can share, of course, with you. That is recently submitted for this. But the question was, the problem, what I'm trying to tell you is that we could figure out the extent of herbivory, but we could not figure out, there are different types of herbivore damages, which precise damage would have been there. So if there would have been better, say, apps or the AI tools with us available, I at least was not aware of. Probably I could have explored and asked the collaborators to use those. But maybe learning from this workshop is probably we are now going to consider them. How best we can have the extent of herbivore damage also assessed from these herbarium specimens. And then we can ask very novel questions in that context. So for example, if we are figuring out the specific traits, see how those traits are linked to different ecological phenomena, like say, for example, say pollination syndromes, right? So all these tools that I have listed there, these are some of the AI tools which are being used in this context in different ways. Now, since, as I said, we work with invasive species, in the invasions, right from the introduction to the complete invasion, we have all these phases where we have the possibility of using one or the other tools, uh, be it management of the pathways of introduction, facilitating phylogeographic approach, of course, or for that matter, even the assessment of the ecological impacts in terms of having that drone data or the remote sensing data or the satellite data, or for that matter, the control. I sometimes was wondering from this TRI database, if you plot somehow the trait variations of aggressive world invasives versus the other extreme, because there are two extremes of species, rare and the most abundant, and how that trait difference would tell you that which are the specific precise traits that would actually predispose species to the invasion, right? So these are the merits of this, I would say, uh, this kind of data, better data reading. And something that of late we have been interested is, since climate change is something that we all now see is a hard reality, and there is enough scientific evidence that yes, it is there, and you in daily life also see a number of kind of very clear signs that yes, it's happening. As botanists, as plant scientists, we see that one of the easiest, the issue basically with this is we don't have the past data. But how can we reconstruct that data? Or how can we peep into the past? So one possibility is, since we look at the plant phenological shifts, because there is now a consensus that there has been almost 1 month, say, prepudement or early spring flowering of many species because of the climate change, driven by the climate change, right? And is it possible for us to go back and move also predict forwards? So what we actually thought, we have been monitoring a bunch of, say, I would say more than 200 species in our botanical garden for the last 5 years now. We have very meticulously collected phenology data for more than 200 species. So both for the functional traits and the phenology. Since when invasion is somehow at your back of mind, you always think from that perspective. We wondered, if there are native versus non-native species, how do they differ in their phenological shifts or functional traits? The idea is that if, for example, climate change creates a niche, a specialized niche, right? There are two possibilities: that this niche would be occupied by a native species, if it is around, or if it is competitive enough, or if it is not, then there's every possibility it could be done by the non-native species, provided it has that advantage of starting earlier. They are otherwise competitive and they have other advantages there. So it is in this context we try to look at a bunch of species, and coincidentally, while we were doing this work, we got a message from one of the collaborators from Germany that there is a network that's working on this phenology that's called PhenObs. So I found that they have a good data set across botanical gardens in Europe and some gardens in North America as well. Once we looked at that data, there were 13 botanical gardens out in 7 countries that we tried to see. We had data of functional traits and the phenology. We asked, say, if native and non-native species differ in their functional traits and the phenology. A data, 440 perennial species we studied for this. And if you look at this trend, we expected actually that these non-native species are probably smarter in terms of they will be starting earlier, and that's how they will be taking the first advantage. And then they will have more protracted sort of cycle during the year. Flowering would be extended. That was our hypothesis. But contrary to our hypothesis, we found something very different. I mean, if you look at this data, native species started much earlier than the non-native species. Then we thought, I mean, how it is so? In our Himalayan context, for example, we thought, yes, this is a smart strategy for the non-native species. Why? Because if they start earlier, there's every likelihood that they can come across many problems like frost, for example. This frost can kill them. They start later, and then they manage to basically finish their life cycle within that time period. And even natives, of course, flowered earlier, with longer flowering duration and fruiting duration, which was not the case with the, non-natives that we expected as such. And then, if you look at the differences in the functional traits also, that was also a bit surprising. We had native species, look at this leaf area, SLA, you know, very very high in case of natives as compared to the non-natives. So which basically thought, what I'm trying to tell you actually is that we found that natives and non-natives as a bunch of species, when you check them at the global scale, they differ. But what limits, before talking about the limitations, we also try to understand the factors that lead to the, prediction of these. For example, exactly where the garden is located, that is a very important predictor. You can see leaf area is a very important predictor for that matter, and so is also the plant height and the others. But recently, one of the colleagues who is part of that network, FinOps, Richard Primick, and some of his colleagues, they came up with a very nice paper in Nymphytologist. This is just a few months back again, where they say that how it is possible for us to use AI tools to scale up these kinds of studies. And there is absolutely huge potential for us to use this herbarium specimen data, which is now becoming more and more available and accessible in terms of its digitization. And there are lessons learned from all cross-continental studies. They have given those examples. And what's very important actually is that since we do not have, it's always a limited problem. When you try to look at a global scale, getting folks to work across places is a challenge. But if the herbarium data is easily accessible to you, you can figure out the phenology and the functional trait data from those herbarium specimens and then see what's happening. So what our effort was in that earlier study, they say could be scaled up multiple times using these different tools that are available now in this very interesting area. Now, in our case, what we are trying to do is as I said, we have a botanical garden in the university at the university campus, which is this 1600 meters elevation. And we are fortunate to also have another botanical garden in Gulmarg, which is at 2600. And then we have another elevational site, which we have, of course, that's not the part of the system. We established it. It's 3,500 meters. So we have established an elevational gradient. Along this elevational gradient, what we are trying to do is we are trying to monitor the same species. We have 35 species that grow in low elevation and the same species at the higher elevation. And if you look at how these species are unfolding their phenology, since in the because of the time constraint, I cannot share that data, but that's really amazing. It's a challenge at times to work in the field in the higher elevation sites 24/7 or more regularly, weekly basis. So what we did was we established these phenocamps at these 1600 meters, 2600 meters, and 3000 meters, these phenocamps. Yes. They take photographs. No, there are different versions of phenocamps. One of the versions, yes. Ours is doing pictures only, but there are other phenocamps that this network is doing. This, yeah, there are possibilities now. So they are high end and they are more expensive than these, of course. But that's quite a possibility of recording the videos. You program it. So what I'm trying to tell you is these phenocamps are programmed for last 3 years now. They are there instantly after every 10 minutes you get a picture. It's a huge data, right? And sometimes I say, ask myself, what the hell this is? I mean, what to do with these pictures, right? And how to basically, you can exactly see at the high elevation site when the snow actually retreats, when does exactly it start, and then how does it differ versus the low elevation sites. And then good thing what we did in the last few years was that we also, along this elevation, after every 200 meters of elevation, we also installed these data loggers, TMS loggers, sensors, data sensors, and getting data on 4 key parameters: soil temperature, air temperature, soil moisture, which is very important. And it's an amazing data set. So I must tell you, we have not touched this part of data yet for the analysis. And I would really be happy if any one of you who is good with this, type of image analysis is interested, we'll be more than happy to collaborate. So my point is that good thing is that it's a real-time monitoring data over the years. And we have followed it with intensive ground truthing. Whatever is possible, normally every week or a couple of weeks after, we go to the sites and we record the data. And there are the possibilities. There are different AI-assisted image processing tools that I believe could be used to make a lot better sense of the data that it must be kind of hiding, be it change in the leaf color, be it change in the flowering, when exactly the bud opens, and how long the fruits persist. Sometimes even in phenological studies, this in our temperate areas, plants shed their leaves. But there are some plants that retain these leaves, what we call as marcescence, right? So why is it so that some species are marcescent, and which advantages this marcescence actually imparts to the species to, deal with the particular environmental situations there. And then, so and then there are possibilities that they say over a century-old data we can figure out through these tools, if it is possible. And through that effort, what is possible actually is how this industrialization, urbanization that has happened over the period of time has actually changed the phenology and the functional traits of the species, right? So having said that, and we also, in this phenology project, what we did was we thought we did a workshop with the college teachers. Actually, it was at ANRF SERP grant. Normally there's a requirement. I must tell you, it was, to begin we thought I could activity kar denge, but it turned out to be very meaningful. The people we got, we developed this manual. It's a complete manual of plant phenology monitoring, and we distributed to them that this is how you can do it. And then we developed an app also, and asked them that we need not to put in a lot of effort since we are working in different institutions or different places. If you can use this app and give us this data. And of late, we figure out that this crowdsource data, of course, with its own limitations, if coupled with these AI tools, can serve a great purpose so far our conservation efforts are concerned in different ways. And there are many tools that are already there, say iNaturalist, WildMe, so on and so forth, which can be basically leveraged upon. So then, since we have now this 5-year phenology data and functional trait data, one of the most important fallouts of this shifting phenology is the pollinator mismatch or match, right? Now, because if we say that plants have shifted their flowering, we have now credible evidence that plants have shifted their flowering sizably, flowering time. Now plant flowers at a time when the pollinator is not actually tuned to be there. So one of the PhD students, Ifa, who is just there, she said that I would like to understand this, and she is now collecting data for last 2-3 years now. This is her 3rd year beginning. Monitoring a bunch of species. She has identified different pollinators, of course. But the issue is, this is again where our basically hands are stuck. We don't have the past data. Actually, we can talk about the mismatch only if you have the past data कि भाई 10 साल पहले 15 साल 20 साल पहले ऐसा था, now it is this way, right? And in that context, there are now some tools that would tell you, for example, this herbarium data, and there are literature which is available on the species, on different pollinator these syndromes. So what are the possibilities of shifting? So this is again an open area where we basically would really see if we can make some headway in getting that past data usefully, to our plate for its better use. And there are, as I said, these AI assisted reconstruction efforts of the data with high precision image analysis tools that are there, both for the better pollinator identification and other things. Then, so one thing that I believe as a student of ecology is that we talk about AI models and tools, but you all know it's their limitations as well, right? One of the limitations I understand is that since if we ask ourselves how much of ecological complexity we understand as of now that exists in nature, there has been a kind of rough estimate that it's very minuscule, less than 10%, even around 5%. What exactly exists in nature? And given the approaches that we have been following, traditional science, is it possible for us to ever understand this whole bunch of complexity? But the question is, so therefore, that's what I'm saying. The merit of field biology and hardcore ecology is very important. Until and unless we do not understand, we do not have access to that quality data, because it's all input output, garbage in, garbage output. If AI is not enriched with the good data, you can't expect basically good outcomes. So my point is that, understanding these ecological interactions in the better, nicer way, and feeding and improving the AI models is one of the challenges that I believe your generation, young people, have. What we did was, I'm just giving one of such examples from our site. As I said, in this grassland sites, we also have along the elevation of gradient. In one of the sites, we have these open top chambers also in place. So what we do is we monitor the functional traits, for example, and cover species, richness, biomass data of the community as such. So you have the control plots where you do not do anything, versus you have the plots where you remove insects, you remove mollusks, you remove fungi, and you remove pathogens. And then you see what does this removal mean for the, and then we do this under the open and then the open top chamber conditions. Because open top chamber, you have normally 1.5 to 2 degrees Celsius difference. Rest everything remaining the same. Just trying to see what would happen to these interactions in the context of the climate change. And if you look at this data, you see as compared to the control, wherever you remove whether fungi or mollusks or both of them or all of them, you have an enhanced biomass. That means these things put the biomass under control. And we have the species cover and richness data and all this data with us. I will not give you those details here. But what I'm trying to tell you is that understanding these complex interactions is very important. And fortunately, we also could see that there are some people who are doing across the world such type of stuff. There's a network called BugNet, where people are asking something of similar questions. And this data is going to enrich, I would say, the models in future, hopefully sizably. And while doing this, as I said, these are the grassland sites where grazing is an issue, right? So we did an experiment, fencing versus non-fencing. What is it that grazing is doing to the systems? Is grazing really important? Is it bad, good, whatever it is? And we found that, and this is also along an elevational gradient, we found that yes, grazing is, of course, if you compare the diversity in the fenced versus the the open plots, you have pretty high species diversity in the open in the fenced areas. It's fine. But if you completely remove grazing, I must tell you there are many things that are happening in the system which says that no, you have in the context of that ecology, what we call that as a moderate disturbance hypothesis. You had this grazing is one of the very important, factors that contributes to the grassland systems. And last month only, we had an option of we had an opportunity to interact with, because an NGO approached us that works on the grassland systems and that deals with these tribal communities. And they said that if we would like to, we would like to learn from you whatever you are doing and share whatever. So we had all those community people and their NGO people and students in the same platform, and we went to the tribal, communities, how they are trying to restore these grasslands. So there are different restoration measures that they are doing, but of course not informed well by science. Many issues not working so well. So what we told them was that, I mean, what are the possibilities of, say, doing it better, smarter, and sensibly, more sensibly? And I could see that yes, there are basically now uh, some this apps available, which could we call it as lead to smarter pasture, say, management. I was wondering if any such app basically we somehow try to use on ground, the benefit of those to these tribal communities who depend for their livelihoods on these pastures is really huge. So in this context, what I believe is that we have come to a point where this AI human collaboration is now so profoundly important. And this is important because it can potentially transform the way these biological collections are curated. They are used for realizing the larger challenges that we have across the globe. And but we have to understand that there are a lot of inconsistencies that we have to deal with when you use the herbarium data, when you use the crowdsourced data, whatever. So there's a paper very recently published. They nicely portray it that as human beings we ask questions, but for facilitating the data, answering those questions, you have an AI assistant. And these AI assistants have to be kind of trait specific. A specific assistant would give you the inputs only about the traits. Another would give you about the curation part of it. Third would give you the identification part of it. So if you look at all these aspects, this seems to be the reality of the day. And we as researchers have to be really now, because it's very challenging as a teacher or as a researcher in this era to deal with the students who have probably better, understanding of many things, access to a lot of information that teachers probably do not have. The you can't wish away. I propose quite often in our meetings to the vice chancellor also that you have no choice than to embrace it. Those old buddies who say that we can't do it at this age, I mean, there's no choice because you can't do. I mean, this is something so fundamentally important in terms of its applications. So, but for that, for overcoming those inconsistencies, I believe this field biology and the ecology that we do out in the nature is so important for that matter. And I see, when I look at the global picture, India is not doing bad. It's doing really good. Because if you look at the number of publications that, mention AI or the machine learning, especially from the environmental monitoring perspective, biodiversity and the data biodiversity analysis, India is pretty ahead of many countries. And recently has gone even further. They say that it has surpassed, in 24 June, it surpassed Italy and the UK in terms of those data, which is a good sign. That means it's picking up in the country, and the workshops like this, the efforts like this, can go a long way to basically take it forward. So to conclude with, I would say that yes, this AI human collaboration and integration is very important because it can really boost the ecological research and take it into very new directions. It can give us the leverage of asking very different questions at very large scales. And then also it can help us to model the ecological complexity of the systems at the desired scales so that we can manage our resources better. We can do our conservation things better. But this is something which needs cross-disciplinary approach. It needs basically the lot of empirical rigor, data-driven effort, data proficiency. Data literacy is, I think, a small word, but data proficiency in that sense of the word. And then dealing with these inconsistencies is something very important. So if we do all this, the most important outcome would be that we will be able to take what we call as the data-driven decisions, which would of course make sense. Otherwise, this part of the world has generally, I would say, not done good in many respects because we have not taken decisions based on the hard data. But I believe these ethical considerations, whatever are there, they need to be taken note of. That's very important. And to just half a minute, this reminds me of something. Long back in probably Cambridge only, there was, I saw it, it was a nature sort of that small piece. There was a workshop in which they had invited somebody who was very old, 80 years or so, to chair that workshop. And he was sharing his experiences when there was no internet in his age versus when there was internet. How did it change the research landscape? But now I believe that when there was no AI, when we were part of it, and now when it AI, when you are now going to be the key players, so what is the difference? So that is something for us to understand very well. And all this good data that I talked about is taught done by me, by all these good students and some of the collaborators and the funding agencies. I thank them. I thank Dr. Gitanjali and the organizers for giving me an opportunity. It was a pleasure to be here. We'll be happy to answer any questions. I hope I'm in time, right?
Thank you, sir, for that informative and insightful session.
Thank you, Manzoor. Brilliant talk as always. And we had so wanted you to be there yesterday, so you could have talked to the MOEFCC director who was here. And of course, but you guys work with the MOEFCC quite often, I suppose. So what I was wondering was, a couple of years back, we had such a huge focus through the Royal Society on herbarium-based research. And the entire Royal Society started giving projects to students, teachers, colleges. Find out a question, use the herbarium specimen and make use of that in different botanical gardens. So they started paying all over the world also small projects. And so in India, the Botanical Survey can probably do something equivalent to what the Royal Society does. And we have so many, as you have said, herbarium specimen all over India. What I found was that you are using them in so many different ways. My question is, we are trying to do herbarium-based genomics. But the problem is genomics may be not for invasive species, let us say. The best you can do is take a small piece, and herbaria are very happy to let you take a sample, then sequence it. But the differences that we have found between native and non-native traits is actually not in the genome, but in the transcriptome. Basically, what part of the genome is getting expressed. And there is no way a herbarium specimen से आप transcriptome तो निकाल ही नहीं सकते because वो तो fix करके रखा हुआ है हमेशा से. So I don't see any way that herbarium can really help in the transcriptomics. But is there a comparative time-based analysis that can be done in your mind? I'm just asking if
it's a very pertinent question. I absolutely got it. I mean, that's the limitation of working with the herbarium specimens, of course. But at the same time, what I see is at least the herbarium specimen would tell you the location of this species at a particular point in time. If not now, I mean, if it is not an extinct species, then there is a possibility that some of the locations it can be there.
So species distribution, it can tell you based on geography.
Yes.
Distribution.
You can at least have a pinpointed, since those days they might not have given the lat longs, but if you have now the place name, you can easily figure it out. And then if you give it a try and figure out that specimen is there, you go to the field and get it. That would be the herbarium informed sort of sampling effort, that can precisely help you in
It's a
doing this.
good idea.
That in the
And get the
procurement.
transcriptome for that
Transcriptomes.
particular
Yes.
sample from that particular region where it came from. And of course, the transcriptome can show geographical changes also in the traits that are there between native and non-native, as we have been seeing.
Yeah, absolutely. I mean, we struggled with this idea. For example, with a species called Coniza,
Yeah.
we have a lot of information globally how it deals with the herbivores, how it deals with the mycorrhizas, how it interacts with the species across that global.
Yeah.
We have this population genetic stuff already done
Yeah.
across the global, so it's fine. Then we thought exactly this: uh, if there are epigenetic variations,
Huh.
and for that you need transcriptomics,
Yes.
right? But how to get this sample from across the range? It was a challenge. So we tried to figure it out. Say some people will do it there using the same method, some will do it here. It didn't work. But somehow I established recently a global population.
Okay.
One is in Germany.
Which you were talking about. Huh.
It is also a possibility. But the issue is that once you bring them in the same environment, then those epigenetic.
Yeah, you're losing the
compromise.
traits that.
But what you are asking is that part can still be answered through that approach.
Through the
I mean, these are the
transcriptome
possible
analysis.
approaches that I personally see are there.
I was also wondering, because you guys being university professors and teachers, you all have access to these students whom you can send everywhere and get things done. Hum log in research institutions, we don't have students per se.
Collaborate with us.
Would love to. I think it's good. Absolutely. Because as teachers, the biggest thing you have is human resource and interested young people who can get things done. We have to hire, we have to keep for a specific time. But
Yeah.
that is one thing we definitely can take up.
Yes, yes.
In the Botanical Survey of India, how often are you talked about BSI. How deep is your BSI? I mean, how often do you talk to the BSI people, and how open are they to having
I believe,
difficult, man?
I mean, there are now changes happening in
Yeah.
the way they are understanding, but I think it needs a much faster and much, at a larger scale effort, because we have very useful collections across the country in the BSI herbaria, and then many institutional herbaria also, which are not necessarily the part of the BSI. So, but still, I believe that there are, there is these generational gaps. I think
Yeah.
The next generation is going to take over these organizations. Probably it would be a better deal. But I'm still hopeful that after my conversations in last few years with the BSI, you know, directors,
Yes.
I mean, there are some
They're positive minded.
some efforts, and they have started doing it, which is basically a good sign.
The BSI director had agreed to come for this workshop. And then she said, "ठीक है, 24th pakka." But she thought 24th April and I thought 24th March. और पूरा का पूरा dates mess हो गया. Now she's willing to
So
come
I
on
messed
24th
up it
April.
for a
तब कौन
day,
आएगा?
messed up it for a month.
So it happens. So in the digital era, we are so messed up. Great, it can happen. But no more questions from my side. Great talk. If anybody else has a question.
Yes, yes. Right. Good. It's a good question. So one that you have many records of the flowering time of many of the species in the literature. That's one source. Second is this herbarium specimens. Once you look at, for example, you are interested in a particular species, you go to the herbarium and you have, say, 50 years back specimen. And then you see, at this date, if there is a specimen, is it with the flower or not? That's another way of doing it. So these are the two, only ways that we have as of now. That's why I was wondering how these AI tools can help us to dive into the past, because you can now reconstruct the climate. For example, climatic history, you can reconstruct climatic, you have two types of phenology. One is flowering phenology, another is, say, for example, this dendrochronology is also a phenology, right? You can reconstruct the climatic history of Himalayas. One of the students who worked with us for masters, but did his PhD with Sukumar at CES, he reconstructed the climatic history of the Himalayas by doing this dendro stuff. So through those approaches, you can figure out what was there, say,